Let's see. Can somebody confirm you can hear me?
I can hear you.
I can hear you.
I can hear you.
Thank you. I just need one person to confirm you can hear me. All right. Before we begin, are there any questions? Let me see if I can see my screen, see if this is working better. Not really. Let me get that a little bit closer. Yeah. Yeah. They won't follow me. There. Well, you can sort of see the background now. Zoom has updated itself, and now I've got to have the green screen behind me to get my background to show up. I'm not sure why that is the case. But it wasn't showing unless I have the green screen there. You can see it's not showing right there. Not a problem for you, just a problem for me. But are there any questions before we begin? Let's share my screen. Let me state that I've only got it one quiz graded, and so far the one student didn't get any points because you need to show me where your computer is sitting. So if you have a laptop, all you need to do that is, I don't know if you can see this. Let me stop sharing my screen. All you need to do is go like this. Show me where your computer is sitting. You can also um, take the monitor and then just go like that. I can't really see very well, but let's see, my hand is here. Well, nope, you want to go further. There you go. And then show me to the left and the right of that. You're going to see my messy desk now. I made my bed now, so I can go that way. I don't know if you can see my bed or not, but you need to do that. Uh, when I say the left and the right, I mean where your computer is sitting, the left and the right. Do not show me the left and the right of you. That would be there and there, which doesn't show me anything. I mean, it shows me the left and the right of you, but I want to see the left and the right of where your computer is sitting. Any questions about any of that? All right, I'll try and get those graded. I wanted to get that graded before class today, and then you would have until the end of tonight, till 11:59, to get the redo the quiz. I might allow you to get the quiz to the environmental quiz. I might allow it to be um, by Friday, like noon Friday or something. Any questions about any of that? No, my background isn't showing. That's the green background screen. Why is that showing? That's not right. I'm gonna have to look into why my background isn't showing. Should be a very nice picture of Clark College there. Um, all right. I'm going to share my screen again. So you should be seeing my screen. Today is July 10th, and we should be finishing chapter 1. I'll try and get started with chapter 2. I doubt we will, but and then there is a lab today. Any questions about any of that? Remember, you have to turn in your environmental quiz. Well, in theory, 11:59 p.m. tonight. The plagiarism quiz is due at 11:59 p.m. Saturday, July 12th. You must get 100% on the plagiarism quiz, or you get 0 points on the 100-point infectious disease project. And then we'll be discussing lab 01 in the lab today. All right, any questions about any of that? If not, I have that we had talked about the beneficial roles that microorganisms play on the planet Earth and to humans. Let's move on to biotechnology and genetic engineering. Biotechnology is the use of microbes and organisms to produce foods and products for human use. Biotechnology is actually centuries old. People think of it as something recent and modern, and it's not. Like the fermenting of various food products like vinegar and, well, I suppose beer, are centuries old. The ancient Romans fermented beer. Genetic engineering is a new technique of biotechnology. And when people think of biotechnology today, they're thinking of genetic engineering, where people are taking the genes out of an organism and then putting it into another organism. Some applications of genetic engineering is we can now produce a variety of proteins from bacteria and fungi. We can use genetic engineering to create gene therapy where a child is born with defective genes. And this is almost always a child is born with defective immune system genes. And then the scientist takes the correct genes into the patient's cells, usually lymphocytes or immune system cells, and puts the genes into them in vitro, and then puts the cells back into the child. And now the child has a functioning immune system. It only lasts for a few months, but the child now has a functioning immune system, and they can routinely um, have the therapy, and then they'll have a functioning immune system, assuming they have the therapy for the rest of their life. And then there's genetically modified bacteria or genetically modified crops, where we can protect crops from insects and freezing by genetically modifying bacteria and then putting the bacteria on the crops. I'm not sure how that works, but I'm sure you've all heard of the genetically modified tomato. It was called the Flavor Saver tomato, which you can't find in the store anymore, but it supposedly lasted longer in the store as a ripe tomato. And so the stores really liked them and they initially had them, but apparently people didn't want to buy them. So uh, they've sort of fallen out of use. Any questions about any of that? One protein that we do make is a human insulin protein. You put it into bacteria like E. coli, grow up the E. coli, and then harvest the insulin from the E. coli, and then give it to the patients as human insulin produced in bacteria. Any questions about any of that? All right, we've talked a little bit about microbes and human welfare. Microbes are extremely important for, well, human welfare as well as extremely important for the planet. Microbes create a microbial ecology. When you think of a person, you normally think of the person, but in reality, the person is a microbial ecosystem, meaning the person has a whole bunch of microbes living in generally symbiosis with the person. Bacteria or microbes create an ecology on the planet. And we already mentioned that bacteria can recycle the nitrogen, allowing the nitrogen to move from one part of the ecosystem to another part. Well, bacteria and microbes can also recycle carbon. allowing carbon to move from one part of the ecosystem to another, as well as there's the sulfur um, geothermal cycle where sulfur is being recycled in the ecosystem. And phosphorus, same thing. Now, it is true that sulfur and phosphorus have a recycling in the ecosystem where microbes are helping it move from one part of the ecosystem to another, changing the form of the nutrient of sulfur or phosphorus from one elemental form to another. But the microbes do not totally create or largely create the sulfur and the phosphorus recycling, meaning water and erosion do release sulfur and phosphorus from rocks. And so that puts a lot of phosphorus and sulfur into the ecosystem, where that doesn't happen so much with nitrogen. And then water and erosion, like wind erosion, can also change the sulfur and phosphorus and move it from one form to another. So that the microbes are not as important in the sulfur and phosphorus recycling in the ecosystem as the microbes are important in the recycling of nitrogen in the ecosystem. And then microbes can also recycle carbon. You all know that when you die, microbes will consume you and then make that carbon in your body available for other organisms, assuming that, you're buried and the coffin you're buried in eventually decays and all that. And then sometime later, the carbon in your body will become another part of the ecosystem, like a tree will put roots down and create um, take up your carbon. So those are examples of the microbial mediated cycles that support life on Earth. We need them in order to recycle the nutrients in the ecosystem. If there was no microbes recycling the nutrients, everything would just accumulate on the forest floor as litter, dead material that wouldn't decay, and the nutrients would not be recycled. The microbial mediated biochemical cycles supporting the life on Earth were actually discovered by Winogradsky and Bajering, and they're the first to show the recycling of nutrients by bacteria in the ecosystem. And they did this from the 1880s to the 1892. One reason why Winogradsky was able to discover this was because when he was studying microbes, he did not study one pure species of microbe at a time. He was studying microbial ecosystems, and to discover the biochemical recycling of nutrients in the ecosystem, you need a lot of different microbes. And so he was studying microbial ecosystems with lots of different species. And he was able to see, he actually found the nitrogen cycle, the sulfur cycle, and one other, maybe the phosphorus cycle. I don't remember. Any questions about any of that? All
No,
right. If you don't recall, I mentioned I do not use the chat. Somebody's using the chat here. I don't see the chats when I'm sharing my screen. So if you have a question you want me to answer, you gotta verbally state it, or you send it to me in an email, and then I'll eventually get back to you with an email. So bioremediation is one way that microbes can improve human welfare. Bioremediation means life, and to remedy, meaning to remedy using living organisms. We can use bioremediation to deal with an environmental problem. Like these men here, they're actually spraying microbes on this oil spill, and the microbes will digest the oil, creating carbon dioxide and water, recycling the nutrients in the oil. We use bioremediation to take care of our sewage. where we use living organisms in the sewage plant to break down our sewage. Bacteria can also break down pollutants like oil here or mercury, and then convert it. If not, the oil can be converted into something that is not harmful to the ecosystem, converted into CO2 and water. Mercury won't be eliminated, so it'll still be toxic, but the bacteria can make it an organic form of mercury, which is less toxic than elemental mercury. Any questions about any of that? We can use microbes to make biological insecticides. Microbes, some of them are pathogenic to insects, and they can be alternatives to chemical pesticides. And they will allow prevention of insect damage to crops and prevent disease transmission, as well as the fact that biological insecticides are less toxic and less harmful to humans as well as other animals than almost all forms of chemical man-made pesticides. An example is Bacillus thuringiensis. That's a bacteria shown here, which you can buy it in a, I don't know, bag there, and then use it to put it on the crops. This bacteria is actually fatal to many insects, and it's harmless to humans and other animals and plants. My father actually bought some Bacillus thuringiensis back in the 1960s and put it on the apple trees. He didn't like it because he only put it on one time and the apples did get worms, although they got less worms than if he didn't put any Bacillus thuringiensis on. And like chemical insecticides, you have to repeat the treatment every 2 weeks, as well as the fact that with Bacillus thuringiensis, you have to put it on after the rain. And he was living in Western Oregon, and we have a lot of rain. So the rain would be washing the Bacillus thuringiensis off the apples. Like I said, he was kind of lazy, he only did it one time, and of course it didn't work. It wouldn't have worked if he used his chemical pesticides if he only used it one time. Any question about any of that? All right. Let's change gears a little bit and start talking about the naming and classifying of microorganisms. Oops, sorry about that. This is the gentleman who established the system of the scientific nomenclature, meaning the naming of organisms, and he also started the classification of organisms. Does anybody know who that is? Or who started the scientific nomenclature and the scientific classification system? You should have had that in Biology 160. Nobody remembers? His name is
What's his name? Carlos?
Linnaeus.
Carlos Linnaeus. Is his Latinized name, and he liked Latin so much he took his own name and Latinized it. His actual name was Carl Linné. That's Linne, I think. And your textbook, I think, calls him Carl Linnaeus, or maybe it's Carlos Linné. I don't remember. But um, meaning they named his preferred Latinized way for one name, and then the other name they kept his real name. So that's not quite correct. So you should really call him Carlos Linnaeus the way he preferred calling himself. But it's true, he was born as Carl Linné. I think he was Swedish, if anyone wants to know. He started the scientific nomenclature where he named each species, each organism, with two names, the genus name and the specific epithet name. So that's not really the species name, because the species will be both the genus and the specific epithet. For example, Homo sapiens is the species for humans. The genus is Homo, which means man who walks on, or ape who walks on 2 feet, and sapiens meaning wise man. Any questions about that? His system was so good for the scientific nomenclature that we still use the naming
Question real
convention
quick.
today. Sorry?
Sorry, I have a question real quick.
Uh-huh.
In the textbook, it says to either underline or italicize the scientific name. Does it matter which one you do?
No. When you're typing, it's probably easiest just to italicize. When you're writing something on the blackboard, it's impossible to write it italicized, so you underline it. But you may do either.
Okay,
I
thank
think
you.
that's italicized right there. Homo sapien. The scientific nomenclature is sometimes called the binomial nomenclature because each species name has 2 names. Binomial means by 2, normal name. So that's actually 2 names and then name because nomenclature sort of stands for name. Any question about any of that? So all scientific names are either italicized or underlined. So if you see it italicized or underlined, you know it's a scientific name. The genus is always capitalized. Coming back to Homo sapien. Oops, I don't need to. It's right here. Let's go forward again. Genus is always capitalized. The specific epithet name is always lowercase. And it's always italicized, shown there, or underlined. 2 other names, E. coli, Escherichia coli is the scientific name. Staphylococcus aureus, also the scientific name. So all scientific names are Latinized, meaning based on Latin, and they're used worldwide. So wherever you go in the world, whatever language they speak, when you say Homo sapien, you mean human. And everybody who knows a scientific name knows that Homo sapien means human. And then Escherichia coli is the bacteria that everyone has in their gut. Staphylococcus aureus, a bacteria that causes skin infection. And my point is, anywhere in the world you go, if you say, I have a Staphylococcus aureus infection, they might not understand I have an infection, because that's in English, but they'll understand Staphylococcus aureus because it's used everywhere in the world. And any questions about that? The name may be descriptive. Orion are a scientist. Homo sapien is descriptive. What it means is wise man who walks on 2 feet. Escherichia coli is both descriptive, coli. It can be found in the colon. And then Escherichia is honoring a scientist. It's named after Dr. Esther, who was an early microbiologist. And then Staphylococcus aureus is descriptive. It literally means golden, aureus, ball or coccus or sphere. It's a golden coccus that grows in a cluster like grapes. Staphylo means grape-like. Coccus, so coccus that grows in a cluster like grapes. Any question about any of that? All right. After the first use, a scientific name may be abbreviated with the first letter of the genus and the specific epithet, meaning the very first time you use Staphylococcus aureus, it should be spelled out, but all other times in the paper, you may abbreviate it as S. aureus. Escherichia coli should be Escherichia coli the first time it's used, but then all other times it can be abbreviated as E. coli. And I will state that E. coli is so common and so well known that even among scientists, they will sometimes use E. coli the very first time they mean Escherichia coli. So this is a bit of an exception because E. coli is better known than Escherichia coli. Any question about any of that? All other times you should spell it out the first time and then write it out. With E. coli, you can do that the very first time. All right, so let's start now turning to the classification of microorganisms. All organisms are classified, and I mentioned that this also was begun by Carolus Linnaeus. However, Carolus Linnaeus classified all living species into an animal or a plant. And as you know, microbes are not really an animal or a plant. And so we do not use his classification scheme anymore. We use a more complicated classification scheme that, I don't know, is a little more correct. We can classify all living things into domains, and there are 3 domains. So all living species can be classified into one of 3 domains. There is the domain bacteria, the domain archaea, also called the archaebacteria, and then the domain eukarya. How the domains are defined is by the genetic data of the species, meaning you can sequence any newly discovered species and then sequence that DNA of that species and then compare it to other species. And you can determine which domain it falls into, because let's say we discover a new bacteria and then we sequence the DNA, its DNA will be more similar to every other species in the domain bacteria than to any other species in either of the other 2 domains, archaea or eukarya. Any questions about that? So the domains are defined, and you just look at the sequence of the DNA and you can determine which domain the species belongs to. The classification underneath a domain is called the kingdom. And unlike the domain, where there's only 3 of them, the kingdoms, it depends on who you are and how many kingdoms there are, meaning different people say there are different numbers of kingdoms. Some people say that there are 3 kingdoms, the same as 3 domains. So kingdom bacteria is also domain bacteria, kingdom archaea is also domain archaea, kingdom eukarya is also domain eukarya. And then some people say there are 5 kingdoms. Most people say there are 6 kingdoms. We'll use the 6 kingdom approach. Other people say there are 9 kingdoms. Other people, 15 kingdoms. Other people, there are 20 kingdoms. And then other people, numerous numbers over 20. And I don't know what they all are, but there are different people who use different numbers of kingdoms, much more than 20. We'll use the 6 kingdom approach because it was the last kingdom approach that was generally used by biologists, as well as it's an easy kingdom to understand, and that is the domain bacteria has only one kingdom, domain bacteria, number kingdom bacteria. Domain archaea only has one kingdom, kingdom archaea. Domain eukarya has 4 kingdoms. Kingdom plantae, the plants, kingdom animalia, the animals, kingdom fungi, the fungus, and then kingdom protista, the protists. Now, what a protist is something that belongs to the domain eukarya, but it is not a plant, not an animal, and not a fungi. So the kingdom protista was always known to be a non-monophyletic group, meaning a collection of organisms that were a eukaryote, but not a plant, not an animal, and not a fungi. And the 9-kingdom approach and the 15-kingdom approach simply divide this one kingdom, Protista, into higher numbers of kingdoms to make 9 kingdoms or to make 15 kingdoms. The 20-kingdom approach starts dividing the bacteria into kingdoms, the archaea into kingdoms, and I think it also divides the plants into 2 kingdoms, as well as dividing the Protista probably into something like, well, at least 9 different kingdoms, if not 15 different kingdoms. Any question about any of that?
The names that are being branched out between bacteria and the topics, are those important for us to know? Because I can't read them on your Zoom.
You can't read this?
No, it's so small because you don't have it like as a slideshow.
I can make it a slideshow, but
Oh,
that
okay.
should be quite easy for you to see. You must have a very strange program that you're not seeing the screen. Can you now read it?
I can read it now, yeah.
Okay. Yeah, you should know that we're going to use the 6-kingdom approach where we divide the eukarya into 4 different kingdoms, Plantae, Animalia, fungi, and Protista. What computer do you have and what system is it using?
I'm using the regular Google Web and I have an iMac.
Google Web. Oh, you're using the
I'm
web.
using Zoom. Yeah, I'm using
You're using
Zoom,
Zoom. Okay.
but I have
You
an
have an
iMac.
iMac. Okay, it must be the iMac that somebody else complained about it being small, and it must be the iMac that makes it
I
small.
also I have a MacBook, and when it's not in the slideshow, the other words are fine, but the ones on the graphic I couldn't read either.
Oh, these right here. Well, this is small. So that I
Yeah.
understand. Yeah.
Yeah,
No, you don't need
that's what I
these.
was talking about. I
No,
can't
you
read
don't
the
need
left side.
these. You don't need to
Oh,
know those. Now, this is showing you that the archaea is divided into 1, 2 different, 3 different kingdoms with, well, that's over 20 different kingdoms right here. I don't know how many it is, but
yeah, I can read your normal
no,
like.
you
Oh,
don't
okay.
need any of this.
Oh,
You don't
okay.
need to know any of that. This you do need to see. Okay. But all of that you don't need to see. All right. Um, the 3-domain system. Remember, the domains is defined. There's no argument about what domain and species belongs to, but the kingdom, there is an argument about what species the kingdom belongs to, and there's also no definition for what a kingdom is. Okay? So just telling you that a kingdom is not defined, and there's a disagreement as to how many different kingdoms there are. Going back to here. Let's now talk about the cell type. Your textbook calls it a cell type. The domain bacteria have the cell type prokaryotes. The domain archaea have the cell type prokaryotes. And then the eukarya have the cell type eukaryotes. Your textbook calls this a cell type, so that's why I'm using it this way. But I will tell you that other authors also call this a superkingdom, something that is bigger than a domain. And there's only 2 superkingdoms, the eukaryotes and then the prokaryotes. And then you divide the prokaryotes into the domain bacteria, archaea, superkingdom, prokaryotes. And then the eukaryotes is not divided from the superkingdom to the domain, meaning it's both a superkingdom and a domain. You don't really need to know the superkingdom. Like I said, I'm just telling it to you because the superkingdom was actually around long before the domains were. People had to sequence the DNA of species before they could get the domains. And that didn't happen until the late 1960s. I think it was the early 70s is when they discovered there were three domains because they'd sequenced enough species and they realized, there's only three domains. But the super kingdom was around at least since the 1950s, if not earlier, which was the cell type prokaryotes versus the cell type eukaryotes. Yeah, let's go on. Some characteristics of the domain bacteria. All the cells are of cell type prokaryotes, meaning they belong to the superkingdom Prokaryota or prokaryotes. They all have peptidoglycan in their cell wall. They mostly multiply by binary fission, where one cell divides into two. For energy use, bacteria can use organic chemicals, get their energy needs, and that's where we get our energy needs from glucose, which actually this is, I don't know if you can see this, but probably not because I'm sharing my screen. A can of soda, but it doesn't really have glucose in it because it's diet or zero sugar. But many bacteria can get their energy from organic chemicals like E. coli. Some bacteria bacteria can get their energy needs from an organic chemical. And that would be a chemosynthetic bacteria. And then some bacteria can get their energy needs from photosynthesis, converting light energy into chemical energy for the cell. Usually the cell will use light energy to make glucose. Most bacteria are either free-living decomposers or are primary producers. So primary producers, something like a photosynthetic or a chemosynthetic bacteria making their own food from carbon dioxide. Some of the bacteria are parasitic and some of the bacteria are pathogenic. Remember, a pathogen would be a bacteria that creates disease, human disease. So a pathogenic bacteria would be one that causes human disease. Of the domain Archaea, they are all cell type prokaryotic. Does anybody know, do any of the Archaea have peptidoglycan in their cells?
No.
You're correct. None of the Archaea have peptidoglycan in their cells. We only find peptidoglycan in the bacteria. Some of the Archaea are found in extreme environments, such as the methanogens, found in environments with methane. The extreme halophile organisms found in extreme salt conditions, like the Dead Sea between Jordan and Palestine, has extreme halophiles, archaea that live in extreme salt. The Dead Sea is about 30% sodium chloride. You can't get higher than that because the salt will precipitate out of the water. And then some of the archaea are extreme thermophiles, organisms that live in high temperatures, like around boiling water or hotter than that. But when we look at all the Earth's biomes, we find archaea in every biome. They are actually a significant number of the microbes in the soil and in the ocean water. What is that called? The plankton has a significant number of archaea in them. Does anybody know if any of the archaea are pathogenic? Do the archaea cause human disease? Anyone have a guess?
They do not.
You guessed correctly. The archaea do not cause any human disease. This class is going to be looking at prokaryote cells and microbes that cause human disease, and none of them belong to the domain archaea. So beyond this lesson, we're not going to talk very much about the domain archaea because none of them cause human disease. And the focus of this class is on microbes causing human disease. I guess We could also say worms causing human disease, as well as microbes. Any question about any of that? So that means that most of the microbes causing human disease belong to the domain bacteria, but there are some fungi, as well as other eukaryotes, protists, that do cause human disease, and we'll talk a little bit about them. And then the helminths, the worms, also cause human disease, and those are animals. Let's move on to some of the characteristics to the domain Eukarya. Remember, the domain Eukarya can be divided into 4 different kingdoms: Kingdom Plantae, Kingdom Animalia, Kingdom Fungi, and Kingdom Protista. All of these kingdoms and all of the Eukarya lack peptidoglycan. So if you find peptidoglycan, it's in the cell wall of a bacteria. Any question about any of that? Kingdom Fungi, that's plural. Singular is fungus. You don't really know to know they have chitin in the cell walls, but you should know that they include the multicellular molds and the multicellular mushrooms. So if you ever look at a mushroom, it's got multiple cells in it. That's why it's visible, by the way. But it also includes the unicellular yeast, which we cannot see with our unaided eye. This would be a mold, and you can see the individual cell right there, composed of filaments called hyphae. So there's one hyphae. Unicellular yeast, where there's one yeast cell. Most of the kingdom fungi are decomposers, but some of them are pathogens. Like Candida albicans is a yeast that can cause, well, skin infections, nail infections, tongue infection, eye infection, a bunch of other infections, but mainly skin infections. Any questions about any of that? The kingdom Protista can be divided into the plant-like protists, the animal-like protists, or the protozoa, and the fungus-like protists. Some of the plant-like protists are the algae, and they're usually green, although they could be brown or reddish. They're photosynthetic, so they use photosynthesis for their energy needs, converting light energy into, well, sugar, converting carbon dioxide into sugar, using light energy. They produce molecular oxygen and other organic compounds like glucose. So they're very important to the ecosystem. The protozoa include amoeba, paramecia, euglena. They absorb or ingest organic chemicals. They may be motile, but if they're motile, they can be motile by flagella, by pseudopods, or by cilia. And this is true of all eukaryotes. They may be motile by pseudopods, cilia, or flagella. For the domain bacteria and archaea, if they are motile, they are only motile if they have flagella, meaning the bacteria are not motile by pseudopods or by cilia. If the bacteria is motile, it's motile because of flagella. Any question about that? Some of the protozoa are parasites, like amoeba can cause amoebic dysentery. That's extremely bad, gut pain and diarrhea, maybe with bloody or mucus in the diarrhea. Oops. Another protozoa, which is not motile, but it is a protozoa, meaning a belonging kingdom protist, is malaria. And then there's fungus-like protists. You'll notice I don't have much there. You don't need to know much about the fungus-like protists. There is one, and that is the potato blight, which caused the potato famine in Ireland in, I think, the late 1800s, which brought a whole bunch of Irish to America because their potatoes were their main crop and they didn't have much potatoes because the potato blight hit the country, and it's caused by a fungus-like protist. Any question about any of that? Let me make sure that's all I wanted to talk about. Yep. All right. For the kingdom Plantae, you can divide them into the mosses, the ferns, the conifers, and the flowering plants. All plants are multicellular. There is no microscopic plant or cellular plant, meaning one-celled plant. Plants do not cause human disease, so we're not going to be talking about them further. Kingdom Animalia includes the animals, the sponges, the worms, and this is actually a misnomer. Helminths is a scientific name for a worm because there are 3 different phyla of worms, and they are as different from each other as they are to humans. Any question about any of that? So we lump them together as worms, but like I said, that's actually kind of a misnomer because the flatworms and the roundworms are as different from each other as they are different from humans. Also in the animals are insects and vertebrates. We're a vertebrate. All animals are multicellular. Some of the animals do cause human disease. Some of the animals are pathogenic, or parasitic is really the term we use for an animal that causes disease. And that would be a parasitic helminth. They're the only animal which causes human, well, I suppose if you had a whole bunch of fleas on yourself, that would be caused by insects, which would suck so much blood out of you, it would possibly cause disease, but generally it's the worms which cause human disease. Any question about any of that? All right, let's talk a little bit about microbiology and microorganisms. What comes under the umbrella of a microorganism? Remember, a microbe and a microorganism are 2 terms that your textbook uses synonymously. I use the term microorganism to mean a microbe, which is an organism. So it has to be cellular. And this is not the way everybody uses the term microorganism, so you don't need to use it that way. I'm just telling you that when I talk about an organism, I'm talking about a cellular organism. And then a virus would be a microbe by my definition, but not a microorganism because it's not an organism because it doesn't have cells. But your textbook does call a virus both a microbe and a microorganism. And that's why you don't need to know either definition of a microorganism. But under the umbrella of a microbe or a microorganism, we have the bacteria, we have the archaea, we have viruses and prions, and these 2 are special because they are not organisms. They do not have cells, but they do cause disease. So we do study them in microbiology, and they are microscopic. Certain of the eukarya are also microbes. The protozoa, the molds and the yeast, some of the algae, if they're microscopic, we call them a microbe. And then the worms, the helminths. But wait a minute, worms aren't microscopic, right? You have a tapeworm that can be something like 30 meters, or maybe it's 30 feet. I used to know. Something like that long. That's not microscopic, right? Everyone can see something that long. Well, worms are studied by microbiologists because microbiologists study things that cause disease, and some worms cause disease, like the tapeworm. Also, another reason for microbiologists to study the helminths, the worms causing disease, is that usually with humans, the infective stage of the worm will be microscopic. So usually with the tapeworm, it's going to be a fertilized egg. It's microscopic, and the fertilized egg is consumed by the human, and then the human gets the tapeworm, which will grow into the tapeworm, which can be something like 30 meters long, which is not microscopic. So of the worms, most of the helminths, not all of them, but most of the helminths are, the adult is not microscopic, but the infective stage or the juvenile stages are microscopic. Any question about any of that? And then I guess something else you can say is that there are a few worms which are microscopic even as an adult, like the adult dog tapeworm is only 3 cells long, and when it makes the 4th cell, 1 cell comes off of the tapeworm, and it has fertilized eggs in it. Any question about any of that? So helminths are multicellular animals, at least the adults. Many of the helminths are parasitic. There are parasitic flatworms and parasitic roundworms causing disease in humans. But the adults are not microorganisms. However, the infective stages are. And some of the other life stages are also microscopic, meaning some of the juvenile stages. And so microbiologists study the helminths largely because they cause human disease. And then the infective stages are microscopic. All right, let's talk briefly about the viruses. We'll continue talking about the viruses near the end of the term. Viruses are acellular. They lack cells. They consist of either DNA or RNA as their core molecule, meaning they either have a genome of DNA or RNA, but never both. It's either one or the other. And viruses are the only genomes that can be RNA. All cells have a genome made of DNA. The core of a virus is then surrounded by a protein coat surrounding the genome. The coat may be enclosed inside a lipid envelope. Not all viruses have a lipid envelope, though. And when do viruses replicate? They only replicate when the virus is inside their host cell. Viruses are not a part of the 3-domain system because they do not have cells. And here we're looking at the HIV virus, this very small red things here. And that's probably a CD4, a human white blood cell, a T lymphocyte right there. Any question about any of that? All right, 3 domains of cellular organisms. Looking at that, we have the domain bacteria, the domain archaea, and then the domain eukaryotes. The cell type for bacteria and archaea is cell type prokaryote. The cell type for the eukaryotes is cell type eukaryote. When we look at their genome, the bacteria generally have one circular chromosome with no histones, no introns. We don't need to talk about the introns now. I'll talk about it much later in the term. By circular, it means the DNA molecule is in a circle, where the beginning point here is the ending point. So generally, bacteria have one circular chromosome. No histone proteins on the chromosome. The archaea also have one circular chromosome generally. And they do have histones. We'll talk about that later. You don't need to know that. Eukaryotes, on the other hand, have generally many chromosomes, like humans have 46 chromosomes. They are linear, like the X chromosome has a beginning point right here and an ending point right there, and those two are not the same point. Any question about that? So eukaryotes tend to have many linear chromosomes, and what differs the eukaryote chromosomes from the bacteria is we have histones as the main protein in our chromosome. And bacteria, they have other proteins, but they do not have a histone protein in the chromosome. The ribosomes of bacteria, they're smaller than the ribosomes of eukaryotes. They're 70S in size for bacteria, as well as archaea. The S just stands for the sedimentation coefficient. That's where the ribosome will sediment in an ultracentrifuge gradient. And then the eukaryotes are a larger ribosome at 80S, and in the ultracentrifuge gradient, they will sediment lower down in the gradient. And the S is just a term for biochemists and molecular biologists for measuring ribosomes. As I've already stated, bacteria have peptidoglycan in their cell wall. So if the bacteria has a cell wall, then you can be sure that they have peptidoglycan as the main molecule in their cell wall. Peptidoglycan is not found in the archaea or in the eukaryotes. Any question about any of that? If not, let's change gear and talk a little bit about a brief history of microbiology. This largely concerns the debate over the spontaneous generation of life and the theory of biogenesis that life comes from other living life forms. Now, when we're talking about the debate about spontaneous generation or biogenesis, you should come to realize that all scientists at all times knew where a baby came from, that a baby never spontaneously generated, with perhaps one exception if you're Christian, but that life comes from life. And even in the one exception, if you're a Christian, you had to say that there was Mary who the baby came from. But normally with scientists, they'd say that a baby always comes from two parents, the father and the mother. So there's no debate about where the baby came from. But other life forms, people thought that the other life forms could spontaneously generate. And this history of microbiology, we'll talk about the debate of spontaneous generation or biogenesis, also concerns the golden age of microbiology, which you don't need to know the year. It was about 1857, after people used microscopes to look at the first microbes, till about 1914. Wherever people looked in the golden age of microbiology, they discovered something new. What's called the golden age of microbiology. Any questions about any of that? All right. So here we're looking at, you don't need to read this, the history of microbiology. And this will be the golden age of microbiology here. This will be the age before microbiology, and then after the golden age of microbiology. And we'll talk about many of these things, not all of them. Any question about any of that? All right, in the history of microbiology, I'm going to discuss things by giving a year But you do not need to know any of these years except for the very first year I give you. And that is when we look at the first living organisms, we can find in the fossil record fossils of the first bacteria or archaea dating back 3.5 billion years ago. Ancestors of the bacteria and archaea that we have fossil records in the Earth's rock. Right there is an actual fossil in the Earth's rock. There's the artist's rendition right there. And let's see if there's anything else I wanted to say. So except for this first, I'm not going to ask you about the dates. However, I may ask you about the relative order of things. So before the Babylonians wrote recipes for making beer, which happened in 4300 BC, you don't need to know this date, but when they made the recipes for making beer, they didn't know it, but they were using microbes to make the beer. And that had to happen after the first life occurred on the planet. And then the next date would be the Mayans make fermented beverage from cacao that we would call similar to chocolate today. They didn't actually make milk chocolate because they didn't have milk, but milk chocolate was actually invented when the Spanish took cacao to Spain when they first discovered America. So that'd be about the 1500s. And I think it was actually Cortez brought cacao to Spain and suggested they put milk in it and sugar, and that's the way hot chocolate came about. So Cortez didn't conquer, who cares? He conquered the Aztecs. You should remember him for the person who invented chocolate. Much more important, in my opinion. Anyways, you should know that before the Mayans could make their fermented beverage from cacao, which used microbes to make that, the Babylonians had to make their first recorded making a beer, microbes making the beer. They didn't know that microbes were involved because they couldn't see them, but they were. And then before that, they had to know that life had to evolve on the planet. So you should know the relative order, but you do not need to know the years. Any question about any of that? You should also know that Janssen developed the compound microscope. You don't need to know when, but before we could look at microbes, we had to have the microscope. And he was the first person to make a compound microscope about 1590. Surprisingly, nobody used the microscope to look at living things until 1665. You don't need to know the year, when Robert Hooke used a compound microscope to look at plants, and he saw little boxes that he called cells in cork. Let me draw this quickly. Well, what happened here? So in a monastery, there'd be like a hallway. We're going to make it this way. And then a hallway going this way. The monks had their own individual rooms that they called cells. And they look like this. And this is what Robert Hooke found when he looked at cork under the microscope. Cells looking like the cells in a monastery. And so he called these cells because it reminded him of the rooms in a monastery. Any question about that? And that's where the term cell comes from, by the way. You should know that when he was looking at cork, he was referring to the bark of the cork tree, which you use that bark to make the cork that you plug like a wine bottle with. And all three terms are correct and mean the same. Well, all cork is used in three different ways. The cork of the bottle, which is the bark of the cork tree, and then of course there's the cork tree. Whenever Robert Hooke looked at plants, he found cells, little boxes. When he looked at animal cells, he did not find, when he looked at animal tissue, sorry, he did not find cells. And that's because his microscopes were blurry and he could not focus in on an animal cell. What do animal cells lack that plant cells have?
A cell wall.
Yeah, the cell walls are thick, and that's why when he looked at plant cells, he could see the cell because of the cell wall being present. When he looked at animal cells, he couldn't see the cell membrane, so he could not see cells. And it needed almost 100 years improvement of microscopes, not quite 100 years, but I don't know, 50 to 75 years improving the microscope before people could look at animal tissue and find animal cells. But his work began the beginning of the cell theory. The cell theory is all living things are composed of cells. It was developed further by other people. Virchow actually wrote it up, the theory that all living things are composed of cells. The above, everything we've talked about, from the development of the microscope, the fossil record on Earth, making of chocolate, is not directly, specifically involved in microbiology, but you needed these things before you could see the first microbe. Robert Hooke surprisingly never found any microbes, but he did find cells in plant cells, or plants. And plant, as you know, is not a microbe. Okay, let's move on. I'll come back to this right here. Let's go on to the first person to look at microbes was actually Antony van Leeuwenhoek. He's pictured there. Let me blow this up. He was the first microbiologist who was such a good microscope builder and lens grinder that he made the best microscopes of his day. He looked, and wherever he looked, he saw microorganisms. He found microorganisms from scrapings from his teeth. He found microorganisms growing in rainwater that had sat for a little while and in peppercorn infusions. And he found lots of microbes in water that he collected from the River Thames, which was the river that the British were in the city of London were drinking for their drinking water. And they didn't really like that they were drinking a bunch of microbes. So he's the first person who saw microbes and then wrote about them. There's his microscope that he actually built. And you'll note that it's a simple microscope. It only has one lens to magnify. And he was such a good lens grinder, as well as a lens microscope builder, that he could see things that other people could not see for nearly 100 years, meaning it took microscopes 100 years later after he made his microscopes, to get microscopes which were a compound microscope that could magnify as well as his simple microscope. A simple microscope, remember, is simply a super duper magnifying lens because it only has one lens to magnify. And he actually ground that lens himself. what he saw were cells right here. This is actually his drawing of cells that he saw. And if you recognize that, these are all cocci. Excuse me, not cocci. These are all bacilli right here, 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 here. There's a bent bacilli. This is a cocci, two cocci, and there's a spirilla. He actually saw bacteria, cells so small that were not seen by other microbiologists for another 100 years. And that's because the microscopes needed a century of development to improve, where the focusing got better, that they got less and less blurry, that people could finally see bacteria. Any question about any of that? All right, let's go back to the theory of spontaneous generation versus the theory of biogenesis. There were two theories to explain how life began. And like I said, I'm talking about simple life forms. I'm not talking about a baby, because nobody ever argued that a baby did not come from the mother and the father. But like a newt, some people argued could spontaneously generate, where life would arise when the vital force came into organic matter, creating the newt. Oh, let me see how I can get this to. So you needed some vital force coming into organic matter, giving rise spontaneously to life. The theory of biogenesis said no, all life arises from another living cell or another organism, or in case of a baby, two organisms, mother and father. So they would say that a newt arises only from another newt. They probably would say about both a male and a female newt, but I don't know. Over a century ago, beliefs included things like you could throw a log into the fire and then a newt would crawl out of the fire. People would say, spontaneous generation of the newt. The wood, the vital organic matter and the fire come together and a vital force comes in, spontaneously generating the newt. In reality, the newt was probably in the wood and it didn't like the heat, and so that's why it crawled out of the fire. Flies could emerge from manure. People would say, spontaneous generation of the flies. The manure provided the organic matter, the vital force came into it, and then spontaneously the flies spontaneously generated. The same with maggots. Most were logical, but none were scientific. Some were even amusing. Somebody wrote up that if you put sweaty underwear in a jar and had wheat kernels in the jar, it would spontaneously give rise to mice. In reality, the mice climbed into the jar to get the wheat, probably started eating the wheat, and then they got trapped in the jar because they couldn't climb out of the jar. The glass was too slick for them to climb out, and the sweaty underwear actually had nothing to do with it. Any question about any of that? We're going to talk about that, but we do need to talk a little bit about that. With microbes, there was a lot of people who believe that microbes spontaneously generate, because when you had a tube of media and then let it sit, it would frequently, if not entirely, become cloudy, teeming with microbes. And people would say they spontaneously generated. In reality, either the media itself had microbes to begin with, or the tube itself had microbes to begin with. And even if you put sterile media and sterilize the tube, microbes would fall into the media from the air. The first evidence starting to disprove spontaneous generation came by Francesco Redi. So you should know Francesco Redi gave the first evidence against spontaneous generation, and you should know him for another reason, and that is he's the first person to write up a controlled scientific experiment. What he did was he got 3 jars, put meat in it, sealed the jars, and those jars resulted in no maggots in the meat. And then he had 3 open jars, and maggots appeared in the meat in the open jars. And people said, no, that doesn't count, Redi, because you sealed the jar. The vital force was not able to come into the meat, and so that's why there are no maggots. And so Redi very ingeniously made a fine wire net or netting, put it over the jar, and then repeated the experiment. He had 3 open jars, 3 jars he completely sealed, and 3 jars that were open, but they were covered with a fine wire net. And the 3 jars that were opened to allow the vital force to come in had no maggots. So this disproved that maggots spontaneously generated, that they only came from the open jars where something came in, laid eggs, it actually was flies, laid eggs in the meat, and then the maggots came in the meat. In the jar with the netting, the flies couldn't come into the meat, and so they had no maggots. So this did not support spontaneous generation, and after Francesco Redi's experiment, everyone agreed that complex living life forms like maggots, anything else complex like a newt, did not spontaneously generate. However, microbes, people still argued, could spontaneously generate. Because if we looked at the meat that was covered with the netting, you would find microbes in the meat. And if you had it in the sealed jar and you sterilized the meat, they would not have the maggots. But if you didn't sterilize the meat and put it in a sterile sealed jar, it would have the microbes as well. So after Redi's day, a lot of people believed that only microbes could spontaneously generate and that higher living life forms could not spontaneously generate. Well, Rudolf Virchow came along and proposed the extension of the cell theory, and he proposed also that living cells can only arise from pre-existing living cells. And this became known as the concept of biogenesis, which had been argued earlier, but he actually wrote it up in the scientific literature and gave it a term, biogenesis, that living things only come from pre-existing living things. This argument continued for a few years. It was not settled until 1861, when Louis Pasteur created an ingenious scientific experiment disproving spontaneous generation and thereby confirming the hypothesis of biogenesis. Louis Pasteur first showed that microorganisms are present in the air, and they're also present in the tube or in the media, and that microbes can be destroyed by heat. And so if you were to sterilize the tube by heating it and the media by heating it, and then sealing the tube, you would not get microbes growing in the tube. But if you allowed the sterile media to have microbes fall from the air, then microbes would grow in the tube. What he did was he then went on to show that spontaneous generation did not happen by his famous and ingenious swan neck flask experiment, where you take a a flask with a very long neck, put the media in, and then you heat the flask so you make a swan neck like that, bending it like that. And then you sterilize the media by heating it. And then you allow the vital force to come in from the outside, come into the organic matter, and you showed there was no microbial growth in the flask unless you tip the media down to here and then move the media back into the flask. What it showed was the microbes fall from the air and then they get stuck in the tube right here by gravity. They don't come up the tube by gravity. And so this would be sterile, would not show any microbes. You can actually find an experiment like this outside the microbiology lab at Clark College, and it's now over 10 years old. It's probably over 15 years old now, where the current chairman of the department made a flask like this and then stuck it outside the microbiology lab, and it's been there for probably about 15 years or more now. No microbes coming here because the microbes are only going there. Eventually, this will dry out, in which case we'll have to toss it away and get a new flask to show. Any question about any of that? All right, well, beginning with Louis Pasteur's work, some other discoveries, advanced science by showing the relationship between microbes and disease. This became known as the germ theory of disease. The germs cause disease. And this was a theory long before it was shown, but we'll talk about the first evidence that a germ actually causes disease. And out of this work came immunity. That is, the immune system can ramp up to fight something, a microbe that's infected the body, and then give you immunity to that microbe so you don't get that disease any further. And then out of that came antimicrobial drugs, that we can find drugs which we can take to cure us of disease by killing off the microbes causing the disease. Like penicillin G is an antimicrobial drug which can cure, among other things, like Staphylococcus aureus infection, assuming the Staphylococcus aureus isn't resistant to penicillin G. Any question about any of that? Let's see if I've mentioned anything. Nope. All right. Got a few more minutes, so let's continue. So during this time, Pasteur developed the basic ideas of aseptic technique, where if you want to study something, you had to purify it down to one species, and then you use aseptic technique to prevent the one pure species from being contaminated by other microbes. And we'll talk a little bit about aseptic technique, like when you have a pure culture, I were to give you, you keep the lid on, you don't allow things to fall down from the air into it. And then when you're taking your cells out of it, for example, you uncover it just briefly, take your cells out, but you need to sterilize the loop beforehand, cool it, grab your cells, and then quickly cover the pure culture so it doesn't become contaminated. That is aseptic technique. As well as the concept of his getting a pure culture, that's not on there. But Louis Pasteur is the first person to develop a technique to getting a pure culture. It was actually a very difficult technique because he had to make dilutions of a liquid, and then he had to make replicas of each dilution. And if ever he found one dilution where one tube had growth and 9 tubes did not have growth, he correctly surmised that the one tube having growth was likely to be formed by one or a few cells and therefore was likely to be pure. Whereas if you had 10 tubes and they all had growth in it, then they were likely not to be pure because the tube probably had more than one cell causing the growth. Any question about that? So he did develop a way of getting a pure culture, but it was not an easy way. He used a liquid culture, taking many different dilutions and then replicas, at least 10 replicas of each dilution. And if one of the tubes came up with growth and 9 of the tubes had no growth, he correctly concluded that the one tube was likely to be formed by a pure culture. Koch developed a technique for getting a much easier technique for developing a pure culture, and that is he streaked for colony isolation on solid media. So you get isolated colonies, and then you take from a pure colony, I mean not a pure colony, an isolated colony, and then you streak it out for colony isolation on a second plate. And if you pass the cells through 2 colony isolation events, then you're likely to get a pure culture. It'll be more than 95% chance that you'll have a pure culture. And that we call streaking for colony isolation to develop a pure culture. So the first time you streak out for colonies, not all of the colonies will be pure. Only about 70% of them will be. 30% of the colonies will not be pure. And that is not good enough in microbiology. Having a failure rate of 30% is not good enough. You want a failure rate 95% or less. And how you do that, you just streak out for isolated colonies on a second plate. Send the cells through 2 colony isolation events. Any question about any of that? All right. About the same time, in fact, 1 year after Dr. Koch developed streaking for colony isolation, in his lab, Hess, who was working in Dr. Koch's lab, developed nutrient agar, a new solid medium for growing many different microbial growth. And that we call nutrient agar. Very simple media for growing many microbes. And then about this time also, Winogradsky developed the concept that microorganisms are involved in the biogeochemical recycling of elements on the planet, such as the nitrogen cycle and the sulfur cycle. Any question about any of that? Like I said, you don't need to know the year, but you should know who discovered which topic. All right. Pasteur then showed that microbes are responsible for fermentation, where when we have fermentation occurring, it's because of the microbes usually converting sugar and fermenting it into something else like alcohol. And that happens with yeast fermenting sugar to make bread, or yeast fermenting grape juice to make wine, or yeast fermenting whatever it is, barley and hops to make beer. So Louis Pasteur showed that microbes were responsible for the fermentation. He also showed that microbes are responsible for the spoilage of food. Like when the beer goes bad, goes sour, it's because of other microbes going in and fermenting the alcohol, breaking down the alcohol into an acid, spoiling the beer. Bacteria use alcohol to make acetic acid, which you can use to make vinegar. Acetic acid is vinegar, making the main flavor ingredient of vinegar, but people making beer did not want the beer to be changed into vinegar. Same with people making wine. They wanted the alcohol. They did not want the wine being changed to vinegar. Pasteur showed that spoilage bacteria could be killed by heat, and the heat was not hot enough to evaporate at least all of the alcohol in the wine. And he invented pasteurization to protect the wine and the beer. Pasteurization is the application of high heat for a short time, used in a wide variety of foods. It's most commonly used today with pasteurizing milk, where you apply high heat for a short period of time to prevent the spoilage of milk. Pasteurized milk lasts longer than regular milk, and then pasteurization is mainly used not to simply give it an extra week shelf life, but also to kill all human pathogens. We'll talk about that later. Oops, sorry. Pasteurization of milk. It's used in a wide variety of foods and medical products today, like some, not antibiotics, but some cosmetics and some medical stuff. I don't remember which, can be pasteurized. All right, the concept of fermentation, that microbes can affect an organic matter, changing it from one form to another, meaning alcohol into vinegar, led to the concept, could microbes have similar relationships in plants and animals? Might microorganisms cause disease in people? This became known as the germ theory of disease. It has no one person who made the germ theory of disease. It came over many different concepts. A lot of people came up with the idea or contributed to the idea. And it was important because the germ theory of disease stated that disease came from a germ, a microbe. And originally, it was believed that poisonous vapors were responsible for at least some infectious diseases, like cholera. They said somebody walking through the swamp would breathe in poisonous vapors, causing the person to get cholera. In reality, the person walking through the swamp got exposed to, oh, I forgot the organisms that causes cholera. Whatever organism causes cholera, Vibrio cholera, I just remembered it. So they'd be walking through the swamp with this organism present. They'd breathe in the organism or get it on their hands and then consume it and then come down with cholera. There were two people who were important for the germ theory of disease. Semmelweis advocated hand washing to prevent the transmission of postpartum or pleural fever between OB-GYN patients. And when he had his doctors wash their hands before delivering a baby, the baby was less likely to come down with disease. And then Joseph Lister used chemical disinfectants to prevent surgical wounds becoming infected. So he'd use chemical disinfectant before surgery, and then greatly enhance the chance that the patient would survive because their wound would not become infected by a microbe. Okay, so both of these people kind of had evidence supporting the theory that germs cause disease. Surprisingly, Semmelweis was fired because the president of the hospital, who was a doctor, was insulted that Semmelweis was suggesting that doctors wash their hands before engaging in medical procedures with their patients. And like I said, Semmelweis was in charge of the OB-GYN, the nursery, so he was having his doctors who were giving birth have his doctors wash his hands. The other doctors in the hospital did not fall under Semmelweis because he couldn't enforce it, but he did suggest it. And the president was so insulted, he fired Semmelweis. Joseph Lister actually used chemicals before surgery and greatly showed that the patients were more likely to survive because their wounds did not become infected. And so he actually continued in his position. Semmelweis did show that the babies were much likely to survive and much less likely to get an infection. He was able to show that, but at his time, nobody cared, or they didn't believe it, or whatever. Robert Koch, and I'm saying that with the English pronunciation. I can't pronounce the German pronunciation. It's something like Koch, because I can't make that sound. But he actually proved the germ theory of disease, where he showed that the bacterium Bacillus anthracis causes the disease anthrax. And he provided the experimental steps required to prove that a specific microbe causes a specific disease, and he published these steps, and they became known as Koch's postulates. The steps one uses to show that a given microbe causes a given disease. This proved the germ theory of disease. And we'll talk about Koch's postulates later in the term, but for now, all you need to know is they're the steps you use to show that a given microbe causes a given disease. Any question about any of that? All right. I want to make sure I wasn't forgetting something. Around this time, Edward Jenner came up with the first vaccine. He made the first vaccine against the disease smallpox. He noticed and heard from milkmaids that an individual who came down with a cowpox infection did not get smallpox. And he initially did not have much evidence from it. He just believed it. And he then injected two boys. One was his own son. The other one, I think, was Edward. I don't remember the other name of the boy. Sorry, drawing a blank here. Anyways, there were two boys that he injected with the cowpox virus, and they got cowpox, which generally is a very mild disease. One in person usually only got one lesion from the cowpox, usually in the site that it was injected into. And then the person became immune to the smallpox virus, and the person was protected against the disease smallpox. So Edward Jenner made the first vaccine against smallpox using the cowpox virus. However, nobody understood why Edward Jenner's vaccine was working. No one understood why a vaccine worked or how they worked. And it was not until Louis Pasteur's work showing that cowpox virus was genetically related to smallpox, so that when the immune system responded to cowpox, some of the immune response, meaning some of the antibodies, could cross-react with smallpox, and therefore make the person not only immune to cowpox, but also immune to smallpox. And then after Louis Pasteur's work, people now understood how vaccines worked. And then people were able to make more vaccines. Like Louis Pasteur made the vaccine against rabies, against a couple of other diseases. I don't remember which. There was a disease against a chicken tumor. It was a virus that causes chicken tumors, tumor in chickens, a cancer in chickens. And he made other vaccinations. I don't remember what they were. And then other people were able to make other vaccines because they knew how to make them. You make it against the agent causing it, either by using the agent or something related to it. And cowpox was related to smallpox. And this protection is called immunity, where the person will become immune to the agent that the vaccine was made to against. And the term vaccine or vaccination is derived from the word vacca. Vacca is the word used for cow, and cow, of course, was the animal that got cowpox, which was a very mild human disease, which nobody was worried about getting cowpox because you didn't die from it. Smallpox, on the other hand, was a very serious disease, and it could kill you. Any question about any of that? All right. So out of this work, we had the birth of modern chemotherapy. Chemotherapy is, in the large sense, the treatment of disease with chemicals. Chemotherapeutic agents against infectious diseases can be synthetic or antibiotics. An antibiotic is a chemical produced by a living organism that inhibits or kills other microbes. This is the textbook definition of an antibiotic. I should warn you that I'm not going to use it that way because the clinicians, don't use it that way. And certainly people in the general public don't use an antibiotic that way. So by this textbook definition, penicillin G is an antibiotic because it is made by the penicillium mold, penicillium mold, and you purify it and then you give it to the patient, and it's penicillin G and protects against bacterial infections, at least some. Most gram-positives, we'll talk about that later. But an antibiotic like ampicillin is not an antibiotic because it is not entirely made by a living organism. Carboxylin would be a better example. It's made in the lab by humans, and so it is a semi-synthetic or a synthetic drug, which is antibacterial, but according to the textbook, it's not an antibiotic because it's not produced by a living organism. I will use the term antibiotic to mean any antibacterial agent, and that's the way it's used in the general public, as well as most clinicians use the term antibiotic that way as well. Let me return to chemotherapy. Chemotherapy today is used almost exclusively for the treatment of cancer, human cancer, by a drug. But it was originally used as the treatment of any disease with a chemical. And it can be used that way, although, like I said, mostly today, we use it and reserve it for treatment of cancer. And so we'll use the term antibacterial or antimicrobial drug instead of a chemotherapeutic drug. Any question about any of that? So Paul Ehrlich happened to study different drugs, and he just tested one drug after the other. And about the 500th drug he tested actually turned out to be an antibacterial drug, which he used to treat syphilis in 1910. He called this a magic bullet, a drug that could be used against bacteria causing infection. The drug was a synthetic arsenic drug, salvarsan, and you could use it to treat syphilis. And he discovered this in 1910. Let's see where we are. The first antibacterial, not antimicrobial drug ever discovered was quinine, which came from the bark of the tree cinchona, or also cinchona. Both pronunciations are correct. And what they did was they took the bark of this tree and then used it to treat malaria. And the bark of this tree had quinine in it, which is an anti-malarial drug. When do you think Europe first had an antimicrobial drug that could be used against malaria? Anyone have a guess? I know you don't know, but just take a guess. Anyone? Well,
1853.
that's not correct, but that's a good guess. It's before then. All right, no guesses. The first use of this bark of the cinchona tree against malaria was recorded in Europe in the mid 1600s. What happened was Jesuit missionaries in Peru noticed that the natives were eating the bark of this tree to treat their malaria, and they noticed that it was successful. And we don't know when the natives first used the bark of this tree, but they took the bark of this tree back into Europe in the mid 1600s and then used it to treat malaria. So that was the first antimicrobial disease. And the exact year is not known, but it was the mid 1600s. And then the year that the natives discovered, nobody knows. It was before that. All right, any question about any of that? The birth of antibiotics came about when Alexander Fleming in 1928 discovered the first antibiotic, and it was penicillin G. What Alexander Fleming did was he streaked out a plate with bacteria, and he was kind of a sloppy microbiologist, and he got a mold colony growing on the plate. And then he left the plate at room temperature and then took off over the weekend on vacation. When he came back to the lab, he found his plate on the counter, and it was growing both bacteria and mold. If he had grown it in the incubator, the temperature would have only allowed one of them to grow, but because he grew it at room temperature, both of them grew. And he noticed something curious around the mold. There was a region around the mold that did not have any bacteria in his streak. And he correctly concluded that something was being secreted by the mold, which is preventing the growth of the bacteria. And he called this drug penicillin. It was actually penicillin G, the first antibiotic discovered. He then experimented with it, but he was only able to get crude media that he could filter out the mold, but that's all he could do. And it wasn't something you could inject into patients, but it could show that the media was antibacterial. The first antibiotic that became clinically available was sulfonamide, a sulfa drug, which was man-made. So somebody made it in the lab, and then they were able to purify it and then inject it into the patients. So it became clinically available in about 1935. So it was the first clinically available, but penicillin G was the first one discovered. And then penicillin became available in World War II in the 1940s, where it was tested clinically. They grew it in mass, purified it, so it's only penicillin G, and then injected it into patients. And if there's ever time, let me tell you the story about the first two patients treated with penicillin G, because it's kind of interesting. But I don't have time today. So sometime we have time, please remind me, and I'll try and tell you the story of the first two patients treated with penicillin G. The second one was actually a good story. Any question about any of that? All right. Bacteria were once classified as plants, hence the term flora. For microbes, you might hear me refer to bacteria as microflora, and that's because that was the term that was used in microbiology for bacteria. It is no longer used, it's inappropriate because bacteria are not plants. They're actually in the domain bacteria. And so the correct term, which is now replaced with microbiota, is the term you should use. You shouldn't use the term microflora. So if you ever hear me say the term microflora, please correct me. I'm pretty good. I don't tend to use that term, but if I do, it's just because I'm falling back to that was the term that was used in microbiology when I took microbiology way back in the 1980s. Tells you how long ago I took microbiology. The normal microbiota are the microbes that are normally present in and on a person. We're talking about the normal human microbiota. Animals and plants are host to symbiotic microbial communities, their normal microbiota, that are central to their welfare. And it is true that they may have some microbes, maybe even many of them, that are the same, but they will differ. For example, the normal microbiota to Felis domesticus, or the kitty cat, includes Bartonella henselae, which are part of the cat's normal microbiota. It is not part of the normal microbiota to humans. And if humans get it, it causes disease, and we call it cat scratch fever. Any question about any of that? So humans have their own normal microbiota. It'll be the microbes growing in and on the person. And it can be similar, like both the cat and the human will have Staphylococcus epidermidis, but the cat has some species like Bartonella henselae that humans do not have. And if humans get that one, they will have cat scratch fever. And don't ask me if there's something that humans have that cats don't have. I'm sure there are, especially in the human gut, but I don't know. Any question about that? The normal microbiota are usually found on and in the skin, the oral nasal cavity, the upper respiratory tract. It's usually not in the lungs, the lower respiratory tract. It is totally throughout the digestive tract and is in the urethra. The normal microbiota is not normally found in internal organs like the blood, the heart, the brain. So it's not usually found in those places. And if it is found in those places, it usually is because of disease. To prevent the growth of pathogens, the normal microbiota can help prevent the growth of these pathogens. They compete for resources. They can compete for space. For example, on my skin, I've got lots of Micrococcus luteus and Staphylococcus epidermidis. If a human pathogen were to fall on my skin, like Staphylococcus aureus, it may fall on top of normal microbiota and then not get established because it has no space to get established. So the normal microbiota can compete for space. It can compete for resources as well. If the normal microbiota eat all the food around the area, even if Staphylococcus falls on the skin, if it has no resources, it can't reproduce. The normal microbiota can secrete toxins that inhibit the growth of pathogens. Yeah, I don't have this just, we'll talk a little bit more about that later, but it can produce toxins that inhibit the growth of pathogens. And so if the pathogen falls near the normal microbiota, it can't get established. The normal microbiota can change the environment, making it so unfavorable for the pathogen to get established, like the skin, the mouth, the urethra. The normal microbiota make all of these environments more acidic. And then a pathogen, a human pathogen that falls in that region may find the pH too extreme for that pathogen to get established. The normal microbiota also produce nutrients and growth factors, which are essential to the host's health. Like in humans, we get some vitamin B12. What else? Folic acid and vitamin K. That's it, vitamin K. Folic acid is vitamin B12. And we get a significant amount of folic acid and vitamin K from our normal microbiota. And then our normal microbiota help us digest our food. Like part of the reason why we can digest the food we eat is because of E. coli in our gut. The reason why a cow is able to eat grass is not because the cow can digest grass, the cow cannot, but it has microbes in its gut that can digest the cellulose, breaking the cellulose, breaking the cellulose into glucose, and then sharing the glucose with the cow. So the reason why the cow can eat grass is because of the microbes in its gut helping digest the food. Any question about any of that? And for us, E. coli helps us digest our food. The normal microbiota do not normally cause disease. However, they can cause disease in certain circumstances, such as when the normal microbiota is growing where it should not be growing. Like E. coli should be growing in the intestines and the colon, meaning the gastrointestinal tract. But if it's growing elsewhere, it can cause disease. Like E. coli growing in the ear can cause an ear infection. In the lungs, it can cause pneumonia. Any question about that? See if I've got any other circumstances. No. All right, you don't need to know about resistance. I think I'm going to finish lecture 1. Let's talk a little bit about infectious disease. Remember, we can classify diseases as an infectious disease and a genetic disease. An infectious disease is a disease caused by a microbe, and infectious diseases can spread from one patient to another. They don't always, but they often do. And we'll talk about what you call one that does, but for now, we're not going to talk about it. So an infectious disease is a disease caused by a microbe, and they may spread from one patient to another, like COVID-19 is an infectious disease that can spread from one patient to another. Staphylococcus aureus is a skin disease and other diseases that can spread from one patient to another. An infectious disease results when a pathogen overcomes the host resistance, grows, and an infectious disease is caused by a pathogenic microbe. A pathogenic microbe, remember, is a microbe that can cause disease. Any questions about infectious disease? All right, let's briefly talk about emerging infectious diseases. An emerging infectious disease is a new or changing disease that is increasing or has the potential to increase in incidence. Can anyone mention a new or changing disease that is currently called an emerging infectious disease?
COVID.
COVID? Is that what you said?
Yeah.
Okay, yeah. COVID-19 is an emerging infectious disease because it's so recent. When was it? 2019 was when it first came out in China. So it's still considered an emerging infectious disease. Can anyone name another emerging infectious disease, one that is not currently blooming and increasing, but they're worried that it might increase in the future?
Is norovirus one?
I'm not sure. I have to look that up.
Maybe influenza because it changes throughout the year.
That
Which-
changes throughout the year, but it's been around for centuries and it does change, meaning it can be a new variant, which then can come out and what do they call that? Causing an epidemic or a pandemic. And surprisingly, we haven't had a pandemic for flu for a long time. The last time was, what was it? The swine flu in something like 2011. Let me do a search there. Oops, didn't mean to show you that. Close that down. Can't do that. Ah,
Would AIDS be one?
you know, I have to look because I'm not sure. AIDS has been around since the 1980s, and so I'm not sure if that's considered new and emerging infectious disease or not, because that's nearly 50 years ago.
What about Zika virus?
That is definitely an emerging infectious disease. Come on.
Lyme disease has been around for a long time too, but I don't know if that's more concerning now in this generation.
I wanted something like just the definition. There we go. Yeah, HIV infections is still considered an emerging infectious disease. Lyme disease is. That has been known since about the 1980s too, if I remember correctly. But that is still called antivirus is still an emerging infectious disease. West Nile virus is because in the Western Hemisphere, it hit the Western Hemisphere, Zika virus is. Let me see if oh. Trying to do this too quickly. "2009, I knew it was a long time ago. The H1N1 swine flu pandemic was the last time we had a flu
Well,
pandemic.
what about the bird flu? Was that after
That
the
was the
swine
one I
flu?
was trying to get at, but let me talk a little bit about H1N1 and then I'll return to the bird flu. Normally, with the flu, about every 10 years, we have an epidemic where the flu changes and people have no immunity to it or very little immunity to it. And so it will be a pandemic that year. And the last time we had it was 2009. That's a little less than 20 years ago. So we're due for a new flu pandemic. Just to tell you. Like I said, I knew it was a long time. And it's like, what is that? 15 years at least. The bird flu was the one I was trying to get at.
I
And
looked it up and the norovirus is currently considered an emerging threat
yeah, it's
by the
currently an emerging
CDC.
infectious disease. Okay. The one I was trying to get at is the bird flu, because it is not one that is currently increasing, but they're worried that it may change and then spread person to person. At the moment, the only way a person can get the bird flu is if they get it from a bird. The bird flu follows the fecal to oral route, and that's the way it's spread among birds, and it doesn't spread respiratory route. And they're worried that if it ever changes and can spread people to people, meaning change from the fecal to oral route, that it can then spread respiratory and person to person. So they're worried that it may change, but
Oh.
it hasn't changed yet. And it's been about 20 years now they've been following it. And so far, it doesn't look like it's going to be changing. So
Now,
hopefully, we
the
don't get a bird flu. A bird
the
flu is
origins
something
of
else.
the Spanish flu,
The
the
Spanish
Spanish.
flu is H1N1, and it has changed a little bit, but it did come out. It actually came out in 1976 and then in 2009. So it's come out in all of those years.
Well, like the original, or like the one of the larger ones was like 1918, 1916, I think.
That was the first flu pandemic that we knew that killed people. Okay.
Now, was it related to?
It's related to H1N1, which came out in 2009, but it's not the
Oh,
same. In 2009, it did not tend to kill people. But other than
so
that, it
that's what
was the
you're.
same H1N1. So it is related to the flu of 1917-18. It's just that it was less lethal.
Ah,
Okay. All right. So you need to know the definition of an emerging infectious disease. That's all you need to know about emerging infectious disease. I have more discussing them, but you won't be tested on it. This is for your own information.
I do have a question about the quiz 1B that we're taking.
Uh-huh.
The study guides that we have for chapter 1 and 2 and 3 is before the quiz 1B. But those study guides are very extensive. So how are we like what like pretty much how am I supposed to know what to really be like focusing on for the quiz?
I'll have to look up to see if you're saying that the material that we're not covering, meaning that quiz one is before the material we cover. I don't think that's right, by the way. Let me just quickly go here. Quiz one is going to cover lab 0, 1, and 2. We definitely have covered those before quiz one. And then it's going to cover chapter 1 and 10. And yeah, we covered that today. So quiz one will not be covering material before the quiz. So that's mistaken. Okay.
So the
For
study
your
guide.
other item, I'm not going to answer that now. Let me send me that in an email and I'll try and make that available to the students. Okay. I think I got a little bit there. Why is grammar up here? Don't know. I think I've got something that'll help. Gosh dang it. Let me stop sharing my screen because I'm trying to grade that. I'm on the grading. Close that down. It's in the announcements, I believe. There we go. Share my screen now. So I have an announcement what to do to get a good grade. Some things that are seen in high-scoring students are they attend class and take notes. The best way to learn something is to learn from your own notes. They review their notes and add to them within a day after taking notes. After a day, your short-term memory is gone. Review my study guide and notes and add them the new information to your notes. Review your notes throughout the term more than just before an exam. Do all the assignments and actively participate. This last is hard to describe, but good students have a good attitude when something bad or difficult comes up. They're willing to work to get it resolved. Poor students have a bad attitude and do little to get the problem solved. So, briefly, I'm only going to talk briefly about Study Guide 1. You should go over the objectives at the very least. Go over the objectives. Turn each objective into a question. If I don't have it a question, I think most of them are questions. And then answer the question. What is a microbe or a microorganism? Should be able to answer that. What is meant by the term pathogenic? Now, this is very specific. There's only one answer for that. But here, know the beneficial role of microbes in recycling nutrients within the ecosystem. There's a lot you can talk about with this one. When something dies in the forest, falls down on the floor, and then microbes recycle the nutrients, breaking the carbon into carbon dioxide and water, making the nutrients available for other living organisms, making the other nutrients available for either themselves, the microbes, and then available for other microorganisms or other organisms, like the potassium in the tree will be broken down so the other organisms can use the potassium. And then recycling of nutrients in the ecosystem, they help recycle the nitrogen in the plant that's fallen down. They'll convert it in from organic matter into urea. The urea can be converted. There's other things you can do. I'm just going to make it simple. Can be converted into nitrite, which can be converted into nitrate. Green plants like nitrate. And then the nitrate can also be converted into nitrogen gas. And then the microbes can convert nitrogen gas into organic nitrogen, like an amino acid. And then that's the recycling of the nitrogen in the ecosystem. And that's only talking about nitrogen. Microbes are also important in the cycling of sulfur, the recycling of phosphorus, and the cycling of carbon in the ecosystem. Any question about any of that? All right, so that's only the beneficial role of recycling of nutrients in the ecosystem. You can then talk about how they're benefiting sewage treatment. We need microbes to break down our waste in the sewage treatment plants. We can use microbes for bioremediation. We're likely spray microbes on an oil spill to degrade the oil and remediate the situation. We can use microbes for food and beverage production. I didn't talk much about that, but obviously we use microbes to make fermented food, like yogurt. Chocolate actually is fermented. Brown tea is fermented. Sauerkraut, etc. We use microbes to make some antibiotics, like penicillin G is made by a microbe. Some pesticides. We use microbes for genetic engineering, like we use microbes, usually a virus, to put the gene in the virus and then put that gene or virus into the patient's own immune system cell, and then put the immune system cell to get the patient now having functioning immune system.
So for that question, if we had something like that on the test, is there going to be more than one right answer?
There would be, but I'm not going to likely give you a essay question. So I will give you a multiple choice question. Almost all of the questions are that, except on the final. In which case, there might be more than one correct answer. Like I might say something like, what is bioremediation? And it would be spraying microbes on oil spill. It could be spraying microbes on a mercury spill. It could be all of the above. And I guess none of the above. And the correct answer for that would be bioremediation would be all of the above. Okay?
Will you get like some points if you answer a couple of them, or it's just 0 if you don't do all of the above?
If you don't do all of the above, this is a multiple choice quiz. It'll be graded by a computer, and so you'll get 0 points. For the historical events, you should know what the discovery or achievement was, why it was important, and what were the scientists were that discovered it or made the advancement. Okay? You do not need to know any years except for the first, and you should kind of know the relative order of the related discoveries. Like vaccines were developed before Koch's postulates. The theory of the germ theory of disease was developed before the theory of Koch's postulate, and then Koch's postulate came out. So Koch's postulate would be last. Any question about any of that? All right. Let's move on to chapter 2. Remember I said I wanted to work it ahead because I don't think you guys are going to want for me to cover the material on Tuesday and then test you on that material on Thursday. So I'm going to try and work ahead. Wait a minute. Do we only have 2 minutes left? I don't remember. Oh gosh, we went way past. You guys should have told me. Class ended at 6:50.
I was gonna say something, but I didn't know if you're.
Yeah, I didn't notice that. I was thinking it was going at 7:20. All right, we will begin the lab at, let's give yourself a 10-minute break at 7:30. And that's going to be very late for the lab. Hopefully no one signed in for the lab. I'll try and log in the lab now, but the lab will not begin until 7:30, so you get a 10-minute break.
When they go on break, can you stay so I can ask you a question?
No, I need to take a break as well, but I'll ask the question, answer the question after I talk in the lab. Okay? All right, any other questions?